Okay, so good evening. Welcome to today's lecture. If you have already been a part of this course, you would know that we have been discussing several different topics under the purview of our course titled as Genes, Genomes, and the Epigenome. Just give me a minute to. Yeah, overall, in the entire tenure of the course, we have been discussing a number of different topics. We had started off with our initial discussion on gene structure, function, and regulation. Then we understood the differences between mutations, polymorphisms, and functional genomics. We understood some key differences between sequencing methods used for NGS, which is next generation sequencing, and Sanger sequencing, the more well, not technically primitive, but a slightly older strategy, which was previously used. And after that, we have modified the Sanger sequencing strategy over time to make it more palatable and more relevant for today's techniques. And following that, we moved on to our discussion on next generation sequencing methods. We understood a lot of detail regarding the different amplification strategies for the template, the PCR reaction strategies, the detection strategies, etc. We have discussed that in the first couple of weeks. This I'm showing you the slide for a slightly older class 2 weeks ago because I want to revise a couple of the topics. But apart from that, today we are discussing things for our 11th class where we'll be starting off our discussion on the alignment of sequences which have been of the reads which have been provided by your sequencing instrument so that it can be aligned to the reference genome. So yeah, coming back here, we discussed different aspects of the genome, exome, and the transcriptome. We began our analysis of high throughput sequencing data from this particular class, which is what I'll be revising the topics we have already discussed, and then move on to our topic for the day. And then we took a little bit of a detour, and we moved on to introduction to epigenetics, and explored some of the key concepts. And in the next class, we will be halfway through our course. So we'll brush up all of the topics that we have already covered, and we'll complete our almost like half of the course. We'll be completing our discussion on sequence alignment by the next class, and peak calling, etc. Maybe we'll do it later on. But overall, I would like to complete like halfway of the course. I'd like to host some sort of an exam or some sort of a test so that we can revise all of our discussions. And yeah, just wanted to, I will drop an email regarding that as well because I don't see any of the students having joined today, but that's what we will do. Now, coming back here, okay, for anybody's reference, this is the book I have referred to particularly in all of my presentations till now. This is just one reference book. So it's a very easy to understand, very easy to grasp textbook, which you can use for your own reference. You can refer to others, and it is easily available online as well. So you can procure it from there. Now, just let me schedule that. Just give me a minute. Next Tuesday, we will be hosting an exam. Maybe not for Tuesday, but maybe I'll drop an assignment in the next session. Um, I'll let you guys know over email. Yeah, okay. I was just revising our previous topic before getting into today's topic. And what I had already just mentioned was that because next week we will be halfway through our course, it will be the 12th week. I will brush up all of the topics we have covered till now very quickly. We'll go through all of those slides and all of the presentations. And if there has been any doubt, whoever would be joining can ask. And sometime in the later end of this week, I'll inform you all over email in the next 1 or 2 days. Either we'll have like a short exam or an assignment. It'll be simple, don't worry about it. Like some MCQs and some just that kind of a easy to do test, but it would be better if you can, wrap up your study for the topics by that time. We will cover gene sequence alignment to the human reference genome by next class, along with the overall recap of the course. And we'll take a pause there. And after we have, completed the assignment or the examination, however it can be hosted, in the session after that, we will take up our discussion for further topics. We will do peak calling, etc., in the upcoming classes, but I want to get an idea about how much people have understood because less number of students have joined. It'll not be a very elaborate thing, but it's just a quick brushing up of your own topics. So we'll probably have that next Friday or Saturday. I'll ask once in advance what is feasible, and we'll continue with our discussion in that same class because it'll be a small test of 15 minutes or 20 minutes, something of that sort, and then we can move on ahead with it. So, taking this discussion forward, we had started off with our discussion on NGS methods. We brushed up our topics on how template amplification and sequencing was performed, and we had begun our discussion on data analysis. Um, we initially started off with the discussion that what you get as an output are the raw reads and your .fastq files, which is where you'll start your data pre-processing. And we understood some key terminologies like fragment, read, mates, sequencing depth, the coverage of your sequencing results or of your sequencing machine. Then we just, yeah. Then we moved on to data formats like FASTA format, the most common one, which most of us know, which is the text-based format for nucleotides as well as for protein sequences. And then we distinguished it with FASTQ format, which is what you get as the output of your sequencing project. And the FASTQ format is also a text-based format where it stores that DNA sequence. And it also has a few other parameters. For example, the first line is sort of an identifier for your sequence, followed by the second line, which gives you the actual sequence of your read, the ATGC composition or the arrangement of ATGC sequences, ATGC bases in your sequence, followed by a separator, which is a plus sign in this case. And finally, the Q scores, which are the quality scores for how well your sequencing reaction has actually proceeded. Then we understood how that Q score is determined or is assigned. And for that, we refer to this parameter known as the Fred score, which gives you a readout as to how well that sequencing has occurred. And we also understood how that conversion takes place between decimal, hexadecimal, and the alphanumeric characters, which you get from your from the ASCII data, and that when converted and finally calculated, you get a net Fred score for each of the reads. And higher the Fred score, higher is the quality and lower is the probability of an incorrect base being called. And therefore, a higher Fred score is more desirable. So yeah, that was pretty much what we discussed. Then we understood a few other formats like SAM and BAM. Currently, what we are going to be discussing in today's class and in the next class, because alignment was an elaborate topic that I wanted to cover, in a little more detail in 2 classes. So SAM and BAM are the two formats which are more relevant in that context, where you get, which is the output of your completed alignment. So today we'll be discussing the basics of alignment, like what was the project, where did it begin from, how are we even, how do we figure out what needs to be done with our, pre-processed, chopped, trimmed reads. So that's what will be our focus today. And apart from that, these formats as well, like JFF and GTF, will also come into discussion today. And these are annotation files which help you in tracing back what portion of the genome your actual sequencing read has come from. So these were the formats we discussed. Then came the bed file format, which is which sort of gives you the capacity to see the features of your sequence. So you basically understand the parameters a little better. It has coordinates which show, it gives you the exact coordinates of the sequence. And yeah, the GTF, GFF, and BED formats, they're interval formats. They retain the coordinate positions for a particular region in the genome. And various other information is also retained, which is what we call as features. Following that, we talked about the VCF format, which is the variant call format, which gives you information about the genetic variants which are present at specific positions in a reference genome. So again, the reference genome discussion is something we'll take up today, where we'll understand it in more detail. And it gives you the information that you get from your VCF file, helps you to figure out or basically identify variants compared to that reference genome, which we have been talking about. So it is to identify deviations from the reference genome. Then we looked, I'll not discuss this elaborately because we have spent like almost 1.5 classes on this discussion. So the different parameters of FastQC include basic stats, sequence quality, per base sequence quality, per tile sequence qualities, per sequence quality score, base content, GC content, N content, the length of sequences and the distribution, duplication within sequences, overrepresented sequences, adapter control, all of this. So these were all of the different parameters that we saw. And then we moved on to the discussion for the next class, where after FastQ file processing, which is where we fed our raw FastQ files, and the tool FastQC was able to read that information, decode it, and test it for all of the different parameters that I had just mentioned, and checked whether or not it meets the minimum criteria, or what is basically the quality of that read or of that sequence that I have provided as the input. Now, once that is done, the next job is to improve the quality. So how do we do that? We basically cut off any portion which is not good, which is a contaminant, any junk, and we trim the sequence to only keep high quality sequences that do not, skew the result in any manner. So the second step, therefore, in brief, is the process of trimming our reads. So what do we mean by this trimming process? We had discussed the use of this particular tool, which is called as Trimomatic, in the previous class, which was developed by USA Dell Lab. And their idea was basically to get rid of, the initial idea was basically to get rid of adapter sequences. Now, again, if you remember, when we did our initial discussion about adapters used for template amplification, we understood how important they are and how necessary they are, and why they are present at the ends of these sequences, because that allows for better bridge amplification. So that's what we saw. Following which, we discussed that there are two possible modes, a paired end mode and a single end mode, and the code which, the command line which needs to be used for each of these modes is also somewhat different, so that needs to be kept in mind. So again, I keep reminding you about this, but a paired end sequencing data and paired end analysis is always a lot more reliable than single end data, and we should, if possible, if feasible, and if budget friendly, we should always opt for the paired end sequencing. So yeah. So the paired end mode, it maintains the correspondence of read pairs, uses information in paired reads to better find adapters or any PCR primer fragments or any sort of tags, additional tags which have gotten introduced during the library preparation process. So Trimomatic, it works with FASTQ files using FRED33 or FRED64 quality scores. Again, remember, a higher quality score is more desirable. And this is an example of what kind of happens in this scenario. So you have a read and you have this junk adapter DNA, which is ligated or attached to it. And using a sliding window method, which we discussed later on, what it basically does is it scans that local region and checks whether the region it is exploring or it has explored is a is not a part of the actual genome and is some sort of an additional DNA, whether it is junk, whether it is a contaminant, whether it's an adapter, whatever may be, that needs to be gotten rid of. Now, next we discussed all of the different parameters which can be used in this context. So these are all the outcome of using certain codes or certain parameters which you can utilize, which you'll find online as well as in the manual for the tool that you're utilizing. So under this, the current trimming steps, they include these parameters like Illumina clip, which can remove all the Illumina specific additional sequences, adapter and other sequences from ends of the read. Then sliding window uses the approach I just discussed. It starts scanning at the 5' end and it clips off the read once average quality falls below a threshold. It detects it as junk and gets rid of it. Therefore, it basically improves the quality of the sequence that we're studying. Max info maximizes the value of each read. Leading cuts off bases at the start of the read if they do not pass the threshold. And the same is done towards the end of a read by the command trailing. Then you have crop, which can cut sequences to some desired length. Head crop will only cut it from the beginning of the sequence. Average quality would ask the tool to drop any particular read if the overall average read quality does not meet our specified levels. Um, then we have top red or to Fred 33 and to Fred 64, which convert our quality scores to these higher quality scores. So following that, we understood the workings of Illumina Clip, where it basically tries to identify contaminants and identify adapters. And its main job is to find the correct balance between sensitivity and specificity. Specificity means that you only want junk DNA to be cleaved off and you want all the relevant DNA to be retained. And sensitivity means that all contaminant sequences are removed and nothing is left behind. So it is sensitive in terms of removing junk DNA with great accuracy. And yeah, apart from that, we talked about the fact that there is a certain phenomenon called as read through, whereby you would see that sometimes the sequencer continues sequencing into an adapter, and therefore some partial or full adapter sequences are often found, and therefore its identification becomes a bit of a challenge. So then we talked about the fact that read through occurs on both forward and reverse ends for paired end data. This was another reason why paired end data sets are more important and are more useful as well. So yeah, Trimomatic includes this strategy whereby it uses the palindrome mode and it uses it as additional evidence to get rid of any such data. So this is how we discussed how a Trimomatic does this job. So basically, firstly, if you have a region which completely aligns to your read, then that is a reliable sequence, and it is a standard alignment procedure, some of which we'll discuss today as well. Second scenario is where only some part of the technical sequence is retained at the 3' end of the read, and so only very short alignment is actually feasible. And Yeah, this is the idea. Then C shows how palindrome mode can also detect long read through. So in C, you can see that beyond the sequence also, large portions of the adapter have also been retained. And this is not useful at all. And if you align both of the paired end reads, you would find the region which is not um, a part of the actual sequence, and then that can be gotten rid of. And indeed, the palindrome approach is used to detect some sort of a short contaminant. And yeah, that is what is basically the idea is that you want to align paired end reads and you want to identify the adapter sequence and you want to chop it off. Yeah. Then I don't want to get into all of this detail, but basically, we discussed how the trimming process actually takes place. You have a initial small alignment, which is called as a seed, followed by checking the alignment between the reed and the adapter, which gets scored. And the alignment score is calculated such that every time there's a match, there's a zero point 0.6 increase, and every time there's a mismatch, the score decreases 1/10. So this is how you end up adjusting it. And yeah, for palindromic matches, longer alignments are possible. And Trigonometric is capable and is sensitive enough to identify even very tiny adaptive fragments which might be present. Sliding window we have already discussed. Then Max Info, its job is to perform an adaptive quality trim, which allows or which basically balances the benefits of retaining longer reads against the cost of retaining any erroneous bases. So um, there are certain parameters under this as well for trimming. There is minimal read length, additional read length, and error sensitivity. So again, I think I explained all of this in detail. Maybe we'll move on to today's class. Time has passed by a lot. Then we talked about leading and trailing as well. We talked about crop, red crop, min length, top red, etc. And this was one example which we had taken for just an example code to show how you can use it if you are running it on a Linux system. So this is the command you need to follow, and the order in which you write the command also really matters because that is how the tool will actually read it. So you have to maintain that order, and preferentially you can get this format or this sequence uh, directly from the Trimomatic website as well, where they have shown how you need to um, analyze your data based on whether it is single end or it is a paired end data set. So yeah. And finally, we had just touched upon the idea of the reference genome, but not gone into the detail of it, but we will take it up today. So as part of week 11, we will have some introduction to the reference genome. So what do I mean by introduction? We'll come to it in more detail, and we will just begin our discussion on sequence alignment to the reference genome and the strategies, and we'll discuss the tools and their practical applications later on. So just a minute. So the idea of the reference genome, that's what I wanted to discuss first. So that's what I've shown here as well. The reference genome, what do we mean by the reference genome? This reference genome is a representative sequence which has come as a result of sequencing a large number of human DNA sequences across the world. And all of this was done under the purview of the Human Genome Project, or the HGP. So it was an international, was and is both, but technically the project has been completed. But the Human Genome Project was an international scientific research project with the goal of determining the simple base pairs, the simple nucleotides which make up human DNA, and also identifying, mapping, and sequencing all of the genes of the human genome from a physical and a functional standpoint. Started in 1990, the project ended or was completed in 2003, and it was an immense, a really large collaborative biological project. Now, keep in mind, things did not like hit a rock in 2003. A lot of advancements, a lot of amendments have been made, and it is something that we expect to learn about more in every subsequent decade, because the genome still, some parts of the genome remain a mystery, and the genome is also not static, it is dynamic, so it keeps changing. So we'll see. Okay. So this table, this I have taken from the official site of the Human Genome Project itself. So they basically want to show the timeline, which shows how the Human Genome Project was, the idea was conceived, how it was, um, what were the major milestones along the process. So here you can see in 1990, the Human Genome Project was initially launched. There were programs called as LCs, or Ethical, Legal, Social Implication Programs, that were founded by the government of the United States. Um, First gene that could have been mapped in this context was the BRCA1 gene, which is a known oncogene for breast cancer. Then in 1991, genome centers were established, second generation human genetic map was developed. Then a 5-year plan was made for the Human Genome Project. And yeah, then you can see that the Sanger Center was also a part of it. I have mentioned it in some of the upcoming slides, that which institutes were actually party to this initiative, to this particular initiative. Then the HGP's human genetic mapping goal was achieved. The first physical mapping goal was achieved. First bacterial genome was sequenced, which was Haemophilus influenzae. Um, yeah. Then we saw the first human gene map. Anyway, the first human gene map, or the genome map, was established in 1996. Some pilot projects for human genome sequencing began. Then first archaeal genome was sequenced. So as you can understand, initially people tried with with organisms which have less complex genomes, and whose cells are easily available, and which might not raise as many ethical issues as human cells. Just excuse me a moment, I keep getting a call. Just 2 minutes. Okay, yeah. So, where were we? We were discussing the first human genome project was initiated with organisms whose genomes are easier to sequence, which are shorter, which are less complex, and obviously will not raise a lot of questions. And in parallel, you can see that more and more about the genome was also discovered, whereby in 1865, you can, okay, yeah, this is an expansion of this 1990 to 2003 domain. But apart from that, Gregor Mendel's achievement, his discoveries, then Henry Sturtevant's discovery, the first linear map of genes. Let me zoom in and see if it's visible. Yeah, it's kind of visible. Yeah, before that, Garrod formulated the concept of inborn errors of metabolism, which sowed the ground that disease is caused by some genetic defects. Then this part you might remember we had discussed in 1944. Avery, MacLeod, and McCarthy, they demonstrated that DNA is the genetic material or the hereditary material. Then the double helical structure of DNA was decoded in 1953. Marshall Nirenberg, Hargobind Singh Khurana, and Robert Halley, they determined the genetic code in 1966. Again, this is something to take pride in that Hargobind Singh Khurana was a part of it, but you might not always find his name to be associated with the same, but he was one of the pioneers of this entire field of molecular biology and genetics, and especially decoding the genetic code was an immense achievement. Um, Following that, Stanley Cohen and Herbert Boyer, they were the discoverers of recombinant DNA technology. Basically, they just saw some molecular scissors and they figured out we can chop DNA and we can join it however we want and we can manipulate DNA. So again, that was an immense contribution. Then the Belmont report on the use of human subjects was issued. Then Sanger sequencing came in 1977. The GenBank database. Now, this somewhere around this time, somewhere around the 70s and 80s, people started realizing, or scientists, researchers started realizing that repositories of data need to be made because it's not possible to publish everything and share everything otherwise. Then the first human disease was mapped with DNA markers, which was Huntington's disease. And one of the potential reasons for this was that Huntington's disease has some characteristic glutamine repeats. Um, Basically, it enriches for increased expression of one particular disease causing protein called as Huntingtin. That Huntingtin is encoded or is composed of large stretches of glutamine residues being encoded one after the another. Now, why that happens is that basically there are AAG sequences, which, if I'm not wrong, yeah, it's AAG, which encodes for glutamine. So polymerase at the time of transcription is not very efficient for AAG trinucleotide, and there is a tendency of polymerase slippage in that region. As a result, these repeats keep on getting enhanced and they keep on synthesizing proteins that cannot fold properly, which is now a dangerous protein to have within the system that is continuously accumulating, and it is called as Huntingtin. So yeah, I diverged a little bit from the topic, but just to because we have moved very far away from biology in the past couple of classes and we moved more towards sequencing data analysis and it was getting a little bit redundant. So I just wanted to give you a little bit of a perspective that, we are learning a very small part of the enormity that mankind has already accomplished and eventually aims to accomplish as well. So following that, the first public discussion for sequencing the human genome was brought about in 1984. Then PCR reaction was invented in 1985. Again, an immense contribution by Carey Mullis. A technique, something so revolutionary we cannot imagine research, the world of academia of the world of biology would come to a standstill without this technique in today's time. Something as simple and as brilliant as this probably has not been made since. That's again my personal opinion, but it was a remarkable discovery. Following that, you can see the International Nucleotide Sequence Database Consortium was formed. Um, then muscular dystrophy gene was identified by positional cloning. The first DNA sequencing instrument was developed all in 1986. Um, then in 87, the first generation human genetic map was developed, which of course again was refined further. And yeah, okay, I don't have my pointer. Anyway, then in 1988, East Artificial Chromosome or GAC was developed, and we will discuss about YACs and BACs in just a few more slides. Following which, Human Genome Organization was formed, sequence tag sites mapping concept was established. Then between 1990 and 2003, the Human Genome Project was actually took flight and was accomplished as well. So without going into too much detail about each and every one of these things, I would just want to draw your attention to some of the key things that happened. So, yeah, so we were here where bacterial genome sequencing was carried out. Then plot projects began for the HGP in the United States. The archaeal genome was sequenced, yeast genome was sequenced, mouse genetic mapping was completed. Then, yeah, then E. coli genome was sequenced. Some other initiatives were also taken into consideration. The new 5-year plan for United States was published. Um, C. elegans genome was sequenced. Then human genome sequencing finally began in 1999. And yeah, then in 2000, we have the first version. Um, US President Bill Clinton and UK Prime Minister Tony Blair, they provided free access to genome information. Then the fruit fly genome was sequenced. So as you can see, over time, people, started sequencing more and more complex organisms with more complex genome organization so that information is available. And here you can see also in parallel the kind of journals that these remarkable discoveries got published in because they were beyond imagination. Then, draft version of the human sequence again published in 2001. Then in 2002, the draft version of mouse genome sequence was completed and published. And then the rat sequence. And finally, in 2003, the final finished version of the human genome sequence was obtained. Yeah. Now, on the left, what you see on the left, what you see are the list of institutes that have been a part of this particular project, the HGP. If you will look here, you'll see that we don't see India or Asia specifically mentioned here. Um, One of the possible reasons could be A, because we did not have that much to contribute, or we were not really considered to be on the global map at that point of time. Secondly, the Indian genome sometimes is very diverse and sometimes is so divergent from other populations that arriving at a consensus might be quite complicated. Thirdly, we have a lot of disparity or a lot of racial disparity or racial inequilibrium, disequilibrium compared to other people from our own population. So the diversity within the population itself also is a major problem. Fourth, again, these are, you won't find these directly stated anywhere, but it's just something that you kind of understand and assume the more you work in this field. Fourth is that currently in India, there have been separate independent projects which have been going on for the past couple of decades where people are sequencing the Indian genome. So initially, it was just a 1,000 genome project where the genomes of 1,000 individuals were sequenced from start to scratch, but then it has been incremented over time to accommodate more and more robust data for better consensus sequence analysis. Just to connect my charger. Yeah. So, now, the project, the Human Genome Project, initially was not able to sequence all of the DNA that was found in human cells. Initially, the idea was only to sequence the coding part of the genome, otherwise, what you can refer to as the euchromatic part of the nuclear genome, which makes up around 92%. And again, keep in mind, saying nuclear genome here is also important because the mitochondrial genome and the chloroplast genome for plants also makes a lot of difference. Again, in case of humans, we do not have anything to worry about the chloroplast, but certainly the mitochondrial genome will make a difference in the sequences that are being analyzed. The remaining 7.9 or approximately 8% exists in scattered heterochromatic regions, which we in general consider to be non-coding and which is found in centromeres and telomeres. And these regions are usually more difficult to sequence, and so they were not included as part of the project's original plans. The initial Human Genome Project did not take them into consideration. And then it was considered, the entire project was declared as complete in April 2003. It was the initial draft was published as well in June 2000, and by February 2001, a working draft had been completed. Then in March 2009 came the new revised committee, which was called as the Genome Reference Consortium, or the GRC. This is what we refer to in today's time when it comes to figuring out the reference genome. What did they do? They released a much more accurate version of the human genome that still had some gaps, but it was not as vague or as empty. Now, it might seem like 92% is a lot, but keep in mind that you are losing 8% of an immense number of nucleotides, so you are actually missing out on a lot of information. So, yeah. Um, Again, I would like to also reiterate one more thing, that euchromatic regions and coding regions should not be used interchangeably. Why? Because euchromatic region are those which are not occupied by histones. As a result, they are accessible to the transcriptional machinery. Now, not everything that is transcribed within a living cell will get translated. So the coding and non-coding part are used just for your reference, but um, for accuracy, make sure that euchromatin and heterochromatin are the correct terminologies. It is a region which is open for fragmentation and for sequencing, versus a heterochromatin is where the region is surrounded or is wrapped around histones and therefore not accessible for sequencing. So be a little more careful with the terminology. I used it loosely, but that's not the correct approach. Now, coming back to the text, in May 2020, the GRC also reported 79 unresolved gaps um, with long-range sequencing techniques, and they finally published the sequence of the X chromosome in May 2020. Similarly, the human autosomal chromosome 8 was also sequenced a couple of months later. Then in April 2022, the telomere to telomere T2T consortium published a complete sequence of the non-Y chromosomes, highlighting 8% of the human genome that the HGP had not sequenced. The T2T consortium used this as the reference to identify 2 million additional genomic variants. So imagine just ignoring some part of the genome is, leading to us missing out so much of information. In August 2023, Rai et al. Reported the successful sequencing of previously missing regions of the Y chromosome, finally achieving the full sequencing of all 24 human chromosomes. So in this is something you might also have seen in the news somewhere in 2023, was not very long ago, where people started to study about the Y chromosome in a lot more detail to, see whether or not it has some roles that we were not aware of previously. So basically, the idea that I wanted to convey here was that the genome sequencing project has not been like a straight line. Every couple of years, or sometimes even months for that matter, there have been upgrades, there have been more and more information. And assuming that the stringency of sequencing was high and was maintained, more and more information, more and more accurate information could be gathered and was incorporated. Following this, the current human reference genome was released by GRC, or the Genome Reference Consortium, in 2013, and most recently was patched in 2019. So this reference, it traces its origin to the publicly funded Human Genome Project and has been continuously improved over the past 2 decades. So another thing to keep in mind is that why we are so bothered about all these different versions, etc. Whenever you use any tool, uh, or you publish your results eventually, you have to mention that at the time of sequencing, what have you used as the reference genome? Because there are two versions which are currently active. There is HG19 and HG38. HG38 is the most latest version, but HG19 is actually something that a lot of tools, even in today's times, are still capable of being or are still compatible with HG19 and not with HG38. So that is something you need to keep in mind. Um, it's better to, especially better to go for HG38 if you are interested in some transcription factors, etc., because they have been sequenced uh, even in more with more resolution in HG38. So anyway, you have to basically look at the version of the genome before you proceed with any analysis and before you report that analysis anyway. Now, unlike the competing Celera efforts, so Celera Genomics, they did this independently. They also took up a project that they want to sequence the human genome. And the most modern sequencing projects based on shotgun sequence assembly, the GRC assembly, because it was done several decades ago, was constructed from sequenced bacterial artificial chromosomes or BACs. Now, I'm not sure if you have concepts about BACs, but basically they are sort of um, like vector systems which are capable of um, they are capable of holding very large or very long DNA sequences, and they can be used for sequencing projects. We'll learn about them in more detail just in this slide itself. So these BACs or bacterial artificial chromosomes, they were ordered and oriented along the human genome by radiation hybrids, genetic linkage, fingerprint maps. Um, but yeah, several GRC assembly gaps were unsolvable because of their some structural polymorphisms, incorrect assembly, etc. So what did I say a lot about? Basically, the idea is that when this project was taken up, we didn't have the technology that we have in today's time. So back in the day, what was done was that DNA was much like today, it was fragmented, but the DNA that got cloned into these vector systems was not always with a lot of accuracy, and long portions or large portions of the genome were actually not even sequenced or not sequenced with sufficient depth to be considered correct. But in today's time, we go for long read sequencing approach, or we use more accurate alignment, etc., where we have improved this to a large extent and have minimized the gaps as much as possible. So this is how BACs work, bacterial artificial chromosomes. They hold very large pieces of DNA. Basically, you can clone very large pieces of DNA. They were originally created from part of an unusual plasmid that is present in some bacteria called as the F prime plasmid. What is it called? It's often called as the sex determining plasmid. Now, what do we mean by the sex or the gender of bacteria? We basically mean that that bacteria can either be F positive or F prime or F minus, whereby they are capable of, you know, um, transforming or exchanging DNA among themselves or not at the time of duplication. Horizontal gene transfer may or may not occur. So F prime had been extensively studied, and it was found that it could hold up to 1 million base pairs of DNA from another bacteria. So that gave the first clue that this can work as an excellent vector system. In 1992, Hiroaki Shizuya, he took parts of the F prime plasmid that were important, cleaned it up, and turned it into a vector. These back vectors, which are artificial chromosomes and which are originally derived from bacteria, but are meant to be used as hosts, they are capable of holding up to 350 kilobases of DNA, and they have all of the tools that a vector needs to work properly. Like they have a replication origin, they have antibiotic resistance genes, and they have sites where DNA cloning or the insertion of our clone of our interest DNA of interest can be done very smoothly. Um, yeah, with these vectors, it's possible to study very large genes, and therefore they became an excellent system for study. And the number of clones required to cover the human genome 6 times theoretically dropped from 1.8 billion to around 50 million by more than half. So basically, what was done in that case was that you have your gene of interest, which can be cut out using a restriction enzyme, and then that can be ligated to the bacterial F prime plasmid, which has been cut with restriction enzymes as well. Then this back or bacterial artificial chromosome gets electroporated into E. coli cells. It's a way of transformation. Then they can be plated on a suitable media, which detects whether or not transformation has taken place. And white colonies are then composed of like Z minus cells. So then you can use that for your sequencing project. Currently, the Genome Reference Consortium or the GRC consists of these institutes. You have the Wellcome Sanger Institute, McDonnell Genome Institute at Washington University, EMBLEBI, the European Bioinformatics Institute, NCBI, the National Center for Biotechnology Information, the zebrafish model organism database, and the rat genome database. Now, these are the institutes that are contributing actively to that project. Now, whatever data they have gotten or they are submitting or they are making publicly accessible, how do you, as the public of interest, how do you access it? So, one of the ways in which I actually even showed you a couple of classes ago. It's fine. Basically, one of the ways is by using the UCSC Genome Browser, which is a portal which is maintained by University of California, Santa Cruz. And as you can see that all of this information on the right is also available on that website, where you can see that the browser assembly that is taken into consideration currently is EDGI 38. This is the Genome Consortium etc. These are all information about the sequencing project from which the current human genome browser data is updated from. Now, till now, what have we learned? We have learned that sequencing is done for very large portions of the genome after chopping it into very large fragments and cloning it into these vector systems known as BACs or bacterial artificial chromosomes. And following that, whatever we get out of it is a sea of immense data that we don't know what to do with. Then comes the idea that now we need to make sense of all the data that we have gotten. How do we do that? So the first thing we need to do is that we need to sort that data. So how will you do that? Now, depending upon what samples you have actually sequenced, whether it is human origin, rabbit, mouse, etc., you will have to generate a genome index of your reference genome before you can align your sequencing reads. So usually comprehensive gene annotation on the primary assembly, sequence regions, and the nucleotide sequence are downloaded. Then what you do, you sort that information. What do I mean by that? Basically, all of the information in the reference human genome needs to be sort of categorized into an index. Why? So that when the tool is searching, then it can search topic by topic instead of searching page by page. So I'm just drawing reference to like a book or some textbook that you have. So it narrows down the computational power that you would need for the same output. So Yeah, the genome sequence and annotation files can be downloaded from a lot of different accessible databases. What I've shown you is the UCSC genome browser, but you can also use GenCode, Ensemble, NCBI RefSeq, etc. And once you have the genome sequence and the annotation file, your your reference genome needs to be indexed. Each genome index has to be created by the software tool that you are using for your alignment. Why? So that after the index has been made, and in the second step, now you want to align it based on that index, both of these data should be compatible with each other. The most common tools which are used for alignment, they're called as Star Aligner and Bowtie 2. So, we will discuss these tools in more detail in the next class because I wanted to pause with alignment, with the introduction to alignment today. But anyway, so the most common tools are Star Aligner and Bowtie 2. So, what do these do? The Star Aligner, what is the idea behind it? It is an alignment tool whose job eventually is to completely align all your reads with your reference genome. But before it does so, it needs to sort or index the reference genome as well. While Star is very good at its job, it is something which requires a lot of computational prowess. It requires at least 30 gigabytes of space, of empty space, and of RAM to align to the human or mouse genomes. And you need to create a directory also for the index, then copy genome FASTA and the gene transfer format or the GTF files into this directory. These are some of the examples. Like for this is the example of the kind of code you need to use. You need to write star, which will command your tool to actually run. Then genome generate, the number of threads you use, your genome directory where the index is actually present, your FASTA sequence, your annotation GTF file, and then you run it. And there are a lot of different parameters which you can sort of adjust here. I've also provided the link to the manual for using star, and it is a GitHub link. I had mentioned, I think, GitHub in the previous class, so you can check it out. It might be quite interesting and quite useful. Now, so one question that might, sort of come to your mind is that why are we going through all of this trouble? What is the need for it? So one of the reasons why you need to go through all this so-called haste is because you are making the process very easy. Which process? So before we understand the problem that indexing is solving, let us understand sequence alignment. Now, sequence alignment is a crucial step of downstream analysis of NGS data where millions of sequence DNA fragments or millions of reads have to be aligned with a chosen reference sequence within a reasonable time. But the problem here is to find the correct position in the reference genome from where the read originates. Now, due to the repetitive regions of the genome and the limited length of the reads, ranging from 50 to 150 base pairs, sometimes what might happen is that shorter reads can map at multiple locations in the genome. However, a certain degree of flexibility for differences to the reference genome should also be allowed. So what do you do to solve this problem? That, how do you figure out where all it is mapping? How do you try to minimize errors? What can you do? Indexing comes in handy at this point of time because NGS data analysis generates immense amounts of data. Now imagine if you have five types of conditions that you're comparing after sequencing, and each of them are also in duplicates, and you have to use computational power for each one of the samples along with the duplicates. Then what will happen is that eventually your data will keep on compounding and increase to an immense level. In that case, using indices will allow you to quickly access and quickly match your sequence data. And these indices are either overall generated reads, are generated over all reads, or they are generated over the entire reference genome, depending on what algorithm you've used. Don't Don't worry, we'll be discussing both these algorithms very soon using videos. Now, with the help of these algorithms, it's possible to compare over 100 gigabytes of sequenced DNA with the human reference genome in a couple of hours. And CPU, you need immense computational capacity as well to reduce the time. And you want more accurate results with as little time as possible. Here on the right, you have a clean definition of sequence alignment. So it is a strategy that is used to arrange sequences of DNA, RNA, or protein to identify regions of similarity. And the basic idea is very similar to that of a puzzle. So an alignment with minimal errors like deletions, insertions, or mismatches would be the most optimal, and ideal is that if there is no error, then it's a perfect match. So yeah, that is the idea of sequence alignment. Now, I think this was, yeah, our last slide for today, after which we'll watch some videos and then we can end today's class. Also, I'll try to start the class on time in the next session. I'm sorry I got late. I had to finish some work and then I could come for the class. But yeah, anyway, we will anyway complete these topics and revise the course in the next class. So we'll try to start by 7 so that we can, wrap up in time. So sequence alignment is very commonly used in molecular biology, and the strategies which are used for alignment very commonly are known either as A, global alignment, or B, local alignment. What is global alignment? It is often called as the Needleman-Wunsch algorithm. Local alignment called as the Smith-Waterman algorithm. So statistically, the space for possible solutions is huge, but we want most optimal alignments with most minimum errors like indels or mismatches. So what is done to actually do that? You try to minimize the distance, which means you try to minimize the number of mismatches, insertions, and deletions in an optimal sequence alignment. And how do you do that? You tabulate partial alignment scores in a matrix, and then you come to a conclusion. The other strategy is local alignment, whereby you take two nucleic acid or two protein sequences, and the only difference is that negative scoring matrix cells are set to 0. Now, you might not have understood a lot from here, but we will look at these videos. Just sorry, my system is terrible. Yeah. So our
In this
first
example, we are going to
learning is going to be about Needleman-Wunsch algorithm. How is the scoring done? Again, keep in mind Needleman-Wunsch is for global alignment, Smith-Waterman is for local alignment.
In this example, we are going to align two sequences. To complete this task, we must remember a few simple things.
Again, like I just said, it takes a matrix into consideration where it takes two sequences, scores each match and each mismatch and each indel or each gap in a certain manner. We'll come to it.
First, leave a space on both sides before writing the sequence. Second, we must assign the gap values in both column 1 and row 1. The following step is to complete the matrix. This is the direction for filling it. Next, and most importantly, how do we fill the matrix? To fill out this box, let's call it box number 1. Values will come from 3 directions: top, left, and diagonally. In light of these considerations, we fill in the boxes in the given example. Every match has a value of +1. Every mismatch has a value of -1. And every gap has a value of -2. So, first, we add a gap position for columns and rows, and then we write the desired sequence. Then, by progressively adding the gap penalties, assign the gap values in both the first column and the first row. In this case, the gap penalty is -2, and the first row and column will be filled as follows. Then we must fill in box number 1. This requires us to consider 3 values: top, left, and diagonal. The top and left values represent the gap penalty, while the diagonal value represents match and mismatch. It means that when we take values from the top and left sides, we add a gap penalty to them, but if we take values from the diagonal side, we add a match or mismatch value to them. So, to find the value of box number 1, we start with the top side, which is -2, and we add a gap penalty of -2 for every top movement, so the answer is -4. Then we take the value from the left, which is also -2, and add it to a gap penalty to get -4. Finally, we take the value from the diagonal box, which is 0. As we all know, taking value from the diagonal box leads to the addition of a match or mismatch penalty. In this case, t corresponds to t, so we add 1 to 0 to get 1. Now, from all of these values, we will choose the highest number and place it in box number 1. Similarly, for the next box, we take the value from the top side and add a gap penalty, yielding -6. Next, we take the value from the left side and apply a gap penalty once more, yielding -1. We took value from the diagonal box in the last step. And now we must add match or mismatch value. In this case, t does not correspond to a, so we add the mismatch value, which is -1, to get -3. Fill the entire matrix in this manner to complete the second step of the algorithm. The final step is to track back to determine the best alignment. Backtracking begins in the bottom right corner and moves to the top left corner. If the two letters corresponding to the box are the same, we proceed diagonally. If the letters are not identical, we will proceed to the highest value on the top and left sides. So let's start backtracking. As I mentioned earlier, if the two letters match, then we move diagonally. A matches with A, so we move diagonally. Next, again, G matches with G. Again, move diagonally. C does not match with G, so we move toward the left or top, where we find the highest number. We have 0 on both sides, which means we can move either way. Now, C does not match with the letter A, so we move toward the highest value either on the top or left side. We've got 2 there, so we move toward that. On the second alignment, A does not match T. Again, we move toward the highest value. Now A matches with A, and T matches with T, so in both cases we move diagonally. The possible alignment should be as follows. Don't forget to subscribe for more informative videos and click the bell icon for the most recent ones.
So this was an example which took a very small sequence which was almost completely identical. But imagine that you are doing this for a very long, like not you, but the tool is doing this for very long sequences, for 100 base pair long sequences, and it is trying to globally align the entire sequence against the entire reference genome. Now imagine that magnitude. So in that case, having an index, what it'll do is it'll go and it'll figure out smaller regions with which it can align. But of course, this requires a lot more computational power, which makes it a lot more laborious. But you get more, like, how do I explain it? There's an advantage to it that it basically is able to align very long stretches at the same time. In contrast, if you look at Smith-Waterman,
The Smith-Waterman
really
algorithm is a
understand it.
dynamic programming method for determining.
Again, you don't have to remember all of this in a lot of detail. I just wanted to familiarize you with the algorithm. This is done in courses which are, when you initially learn alignment for protein sequences, when you're learning BLAST, etc., in bioinformatics associated courses. But because our approach is more towards data analysis and we are quite heavily reliant on tools, I'm not going to put a lot of stress into it, but if you're interested, you can obviously read as much as you want from any source that you want. But I just wanted to familiarize you with what the tool, each type of tool which uses one certain sort of algorithm is doing at a, very base level.
Similarity between nucleotide or
Let me
protein.
switch on the subtitle. There are no subtitles. Okay. Yeah.
Programming method. The Smith-Waterman algorithm is a dynamic programming method for determining similarity between nucleotide or protein sequences. The algorithm was first proposed in 1981 by Smith and Waterman and identifies homologous regions between sequences by searching for optimal local alignments. To find the optimal local alignment, a scoring system including a set of specified gap penalties is used. Optimal local alignments are identified by comparing the query sequence with the sequences in the database on a character-to-character level. Contrary to the Needleman-Wunsch algorithm, on which the Smith-Waterman algorithm is built, the Smith-Waterman algorithm searches for local alignments, not global alignments, considering segments of all possible lengths to optimize the similarity measure. The algorithm is based on dynamic programming, which is a general technique used for dividing problems into sub-problems and solving these sub-problems before putting the solutions to each small piece of the problem together for a complete solution covering the entire problem. Like the Needleman-Wunsch algorithm, the Smith-Waterman algorithm has 3 steps. Initialization, matrix filling and trace back. We also have match rewards, mismatch penalties, and gap penalties. Here, we have 2 nucleotide sequences. But first, leave a top row and left column to assign the gap value, then write the given sequences on the top and left sides. Unlike the Needleman-Wunsch algorithm, in the Smith-Waterman algorithm, the top row and left column are filled with 0 to initialize the matrix. The second step is to fill the matrix. To fill the matrix, we considered the maximum value from the 4 cases. First, we consider the value from the top side and apply a gap penalty to it. Second, we take the value from the left side and add a gap penalty to it. Third, the diagonal value is considered. Now, if the 2 letters match, we add a match reward to the diagonal value. But if the 2 letters do not correspond to each other, we add a mismatch penalty to the diagonal value. Finally, in the 4th term, placing a 0 in the recursion ignores a possible negative alignment score. In the Smith-Waterman algorithm, there is no negative value. To fill box number 1, we take the value from the top side, which is 0, and add the gap penalty. Then, take the side value and again add the gap penalty. In the 3rd place, take the diagonal value and add the match reward to get +1. As A corresponds to A, and the last one is 0. Now, consider the maximum number and put it in the box. For the next box, repeat the same procedure for the top and side values, but for the diagonal value, subtract -1. As t does not correspond to A, consider the maximum value from all 4 and put it in box number 2. I hope you get my point. Fill the matrix according to the given instructions. The final step is to trace back. In trace back, start from the highest value anywhere in the matrix. So, the matrix represents a number of local alignments. Don't forget to subscribe for more informative videos and click the bell icon.
So, again, let us bring back some perspective into the discussion. This was a lot of detail. If you're interested in it, you can learn it either from this book. Here also it's given, but I felt it might be a little more confusing. It's better to, go into videos because they're showing it live to you. But the book that I have referred to, it has global alignment, Needleman-Wunsch algorithm. Here, like you saw, the penalties for match, mismatch, and gap have been given. And there's a diagonal jump where you read the final sequence from the bottom to the right, from the bottom right to the diagonal upwards. And you have local alignment as well, which is for Smith-Waterman algorithm, um, where it does a couple of things differently, but let's not get into that. But basically, the way of tracing it is also different, and there are no negatives in a Smith-Waterman matrix. There are different types of tools which use different types of alignments, but yeah, we will not get into right now. We'll start our discussion with the alignment tools of star and Botai2 in our next class. But I just wanted to introduce it to you. You can read more and more about it as much as you want. But overall, what you should know is how the tools work, what sort of strategies tools use to figure out the sequence alignment. Basically, it is doing the same thing, but at an unimaginable scale where it is using computational power to do what a human would take probably days to complete. It's able to do that a lot faster. Secondly, we are splitting the genome into indices to simplify the process where multiple indexes can be checked by multiple parts of your CPU basically at the same time. You can use different threads. If you use a high like there are certain supercomputers also, right, in some institutes, in some research institutions, etc., which have even more computational power, which can be dedicated. But of course, you should be able to install your tools onto those. Your tools should be compatible with the genome version that you have been using. Mostly, these tools are Star as well as Botai 2. Both, I think, both of them are EDGE38 compatible. So that should be the case. For example, if it was EDGE19 compatible, you would not have been able to use it. So there are many ifs and buts in this manner. But yeah, that is pretty much what I wanted to discuss for today. We will take up Star and Botai 2 in the next class, and we'll sum up all our discussions till now. It has been around 130 slides. That's a lot. So of course, I'll not be discussing in as much detail, but we will be brushing up all our topics very quickly so that any doubts on anything you want to discuss, we can pick up that topic and we can wrap up our halfway mark through the course and then we can proceed forward. If you have any doubts, please let me know. Read about whatever topics you find interesting in more detail. This course is to familiarize you with topics. I cannot cover everything, but I am trying as much as possible without making it too boring because I know it's a lot of theory at this point. But yeah, that is all I wanted to discuss for today. Please let me know if you have any doubt, and then we can end today's class.
Ma'am, today's topic was clear to me.
Okay. Have you done this before, the alignment
Yes,
matrices?
yes, ma'am. Yes, ma'am. Yes,
Okay,
ma'am.
okay,
I
cool,
have
cool.
done like
Otherwise, it's a difficult topic to grasp.
ma'am, in
Okay.
this semester I had done this.
Okay, okay, great. Okay, cool. So we'll proceed with the discussion on the alignment tools, and I'll drop an email about whatever we can do. Basically, you and another student were the only one who were joining. So we'll
Yes,
just have like a short discussion on based on questions or something of that sort. I'll drop an email about it. But class will be as usual on Tuesday, and after that we'll have
Ma'am,
some
one thing I
session.
wanted to ask, like you
Yeah.
have mentioned about the test. In that,
Mhm.
the previous two lectures will also be there. Like the questions from previous two lectures will also be there, or it will cover only the starting portion, or because
Um,
those two were like very.
Would it be easier to not include? Yeah, okay, maybe we can keep that topic separate, like from data analysis onwards. Okay, yeah, maybe we can skip it. I'll keep it to the topics before that.
Ma'am,
And
after 7th, like after 7th Feb, you can take that. Like I have to give my exam of GATE, and
Okay, okay.
after my exam, I'll watch that videos, and after that,
Sure,
you
sure,
can
sure. I
take
haven't uploaded them
exams
also.
from
Okay.
for that.
Sure, sure. No problem. I'll
Yes,
upload
ma'am. Yes,
these
ma'am.
as well. And then we can have a like a small MCQ test on the data analysis topics separately. That works.
Done, done, done.
Take. Okay. Thank you for joining. I'll end the
Thank you,
class
ma'am.
now. Okay, good night.